How antidepressants change brain circuits. Antidepressant medication comes in many different forms and are used more widely than ever before in modern society. Today we take a look at the different types of antidepressant and how they interact with the human brain. Let's get into it. 1. Introduction. Antidepressants do more than simply increase neurotransmitter levels. While many medications initially alter the concentration of neurotransmitters such as serotonin, norepinephrine, and dopamine, the longer-term therapeutic effects appear to arise from changes in brain circuits and neuroplasticity. Depression is associated with dysfunction in several interconnected brain networks involved in emotion, reward processing, memory, and stress regulation. Antidepressants gradually modify these circuits through changes in neurotransmitter signaling, receptor sensitivity, gene expression, synaptic plasticity, neural connectivity between brain regions. These adaptations develop over weeks to months, which explains why antidepressants often take several weeks to produce significant clinical improvement. 2. Brain circuits involved in depression. Several key brain regions form networks that regulate mood and emotional processing. Prefrontal cortex, PFC. The prefrontal cortex plays a central role in decision-making, emotional regulation, impulse control, cognitive evaluation of emotional stimuli. In depression, activity in parts of the PFC, particularly the dorsolateral prefrontal cortex, is often reduced. This can contribute to impaired concentration, negative thought patterns, and difficulty regulating emotional responses. Antidepressants help restore more balanced activity in this region, improving cognitive control over negative emotions. Amygdala. The amygdala is involved in detecting emotionally significant stimuli, especially threats. In depression and anxiety disorders, the amygdala often shows hyperactivity, leading to heightened emotional sensitivity, excessive fear or worry, increased response to negative stimuli. Antidepressants reduce excessive amygdala reactivity, which helps decrease emotional over-responsiveness and anxiety. Hippocampus. The hippocampus is critical for memory formation, contextual processing of emotions, regulation of the stress response. Chronic stress and depression are associated with reduced hippocampal volume, partly due to decreased neurogenesis and increased neuronal atrophy. Antidepressants promote growth and repair in this region by increasing neuroplasticity. Reward circuit, mesolimbic system. Motivation and pleasure are controlled largely by the dopamine-based reward pathway, including the ventral tegmental area, VTA, the nucleus accumbens, parts of the prefrontal cortex. Depression often involves reduced activity in this circuit, leading to symptoms such as anhedonia, loss of pleasure, low motivation, reduced energy. Certain antidepressants, particularly those affecting dopamine and norepinephrine, help restore activity in these reward pathways. 3. Immediate versus long-term effects of antidepressants. Immediate neurochemical changes. Within hours of taking an antidepressant, serotonin levels increase in SSRIs. Norepinephrine increases in SNRIs. Dopamine may increase in certain medications. However, these changes alone do not immediately relieve depression. Long-term neural adaptation. Over weeks of treatment, the brain begins to adapt through several mechanisms. Receptor regulation. Neurons adjust the sensitivity and number of receptors responding to neurotransmitters. Examples include downregulation of certain serotonin receptors, increased sensitivity of mood-regulating pathways. These receptor adjustments help stabilize emotional signaling networks. 4. Neuroplasticity and synaptic remodeling. One of the most important long-term effects of antidepressants is the enhancement of neuroplasticity, the brain's ability to change its structure and connections. Brain-derived neurotrophic factor, BDNF. Many antidepressants increase levels of brain-derived neurotrophic factor, BDNF. BDNF is a protein that supports neuron survival, synapse formation, growth of new dendritic connections. Higher BDNF levels strengthen communication between neurons and help repair circuits damaged by chronic stress. Synapse formation. Chronic stress and depression can cause loss of synaptic connections, reduced dendritic branching in neurons. Antidepressants gradually promote formation of new synapses, strengthening of existing neural connections. These changes help restore healthy communication between mood-regulating brain regions. 5. Regulation of stress circuits. Depression is strongly linked to dysfunction in the body's stress response system, particularly the hypothalamic pituitary adrenal (HPA) axis. In many depressed individuals, cortisol, the primary stress hormone, remains chronically elevated. Stress responses become dysregulated. Antidepressants can normalize this system by improving communication between the hippocampus, hypothalamus, and prefrontal cortex. As these circuits stabilize, stress responses become more balanced. 6. Changes in emotional processing. Functional brain imaging studies show that antidepressants begin altering emotional processing before mood improves. For example, after several days of treatment, the brain becomes less reactive to negative emotional stimuli. Positive emotional signals are processed more strongly. This shift in emotional bias may gradually lead to improved mood and behavior over time. 7. Strengthening communication between brain regions. Healthy mood regulation requires coordinated activity between several brain areas. In depression, connectivity between regions such as the prefrontal cortex, the amygdala, the hippocampus may become disrupted. Antidepressants help restore communication between these regions by strengthening synaptic signaling, improving functional connectivity in neural networks. This allows the prefrontal cortex to better regulate emotional responses generated by the amygdala. 8. Differences between antidepressant classes. Different medications influence brain circuits in slightly different ways. SSRIs primarily affect emotional regulation circuits, reduce amygdala hyperactivity, strengthen prefrontal control over emotional responses. SNRIs improve emotional regulation, increase energy and alertness through norepinephrine pathways. Dopamine-based antidepressants enhance reward circuitry, improve motivation and drive. 9. Emerging treatments and rapid circuit changes. Some newer antidepressant treatments appear to work much faster because they directly trigger synaptic remodeling. For example, ketamine-based treatments rapidly increase synapse formation in the prefrontal cortex. Psychedelic-assisted therapies may temporarily disrupt rigid brain networks, allowing new patterns of connectivity. These treatments highlight the growing understanding that depression may involve maladaptive neural network activity, not simply low neurotransmitter levels. 10. Conclusion. Antidepressants alter brain circuits through a multi-step process. Initially, they increase neurotransmitter levels, but their longer-term therapeutic effects arise from deeper biological changes including receptor adaptation, increased neuroplasticity, enhanced synaptic connectivity, improved communication between mood-regulating brain regions. These changes gradually restore balance in neural networks that regulate emotion, motivation, and stress responses. Understanding these circuit-level effects helps explain why antidepressant treatment requires time and why combining medication with psychotherapy, which also modifies neural circuits, can be particularly effective.